session on speciation, and I'm also the first speaker, and I will present a study that is part of the PhD work of Yasmin Latour that was done at the Institute of Evolutionary Sciences in Montpellier. And this study is about testing whether agonistic outer displacement could explain a pattern of associative mating that in the house mouse that we interpreted as the result of a process of reinforcement. So when two sister populations evolve in allopathic conditions during which they accumulate divergence and sometimes incompatibilities, if they meet along the secondary contact, they could interbreed, and if they interbreed and produce maladaptive, they will produce maladaptive hybrids because of the accumulation of incompatibilities. And theory predicts that selection against hybridization would favor the evolution of character displacement in the form of associative mating in the two parental population, and this is what we call reinforcement. However, recently it was proposed that other mechanisms could produce the same pattern, and hence we have to be careful about it. And one of the mechanisms is competitive interference, where the two populations share part of their niches and could compete directly through dominance of one of the sexes. And what was proposed here is that signals involved in mate recognition could evolve in the framework of competitor recognition evolution. So this is what we decided to test after having shown that there might be reinforcement in the house mouse, and I'm going to present you our first result on reinforcement. So our study model is the house mouse, and the house mouse is a wild animal that originated in Asia some 100,000 years ago. It colonized Europe through two independent routes, and during this time it has accumulated divergence, resulting in two subspecies, Musculus, which is the short-tailed house mouse, and Domesticus, which is the long-tailed house mouse. And these two subspecies are called subspecies because they hybridize wherever they meet, and they form a secondary contact going from Scandinavia to the Caspian Sea. And we have been studying more particularly the Danish hybrid zone, which is dated, which is believed to have occurred some 3,000 years ago at the maximum. So the hybrid zone is studied in different parts of its geography, but we have studied this one. So this house mouse hybrid zone is characterized as a unimodal hybrid zone, which means that the hybrids are mainly recombined genomes, and there is some gene flow and introgression by some genes, but there is very strong selection, at least in Denmark, on the sexual chromosomes. We also show that there was selection against hybridization through studies of fertility, showing lower fertility of the hybrids and also lower attractiveness of the hybrids, at least for the parental forms. So this was a good condition to test for reinforcement, and this is what we did. And we compared mate preference in Musculus and in Domesticus, in allopathic populations, and across the hybrid zone, going from the border of the hybrid zone through the zone. And what we found is that there is no preference in an allopathic population for Musculus or Domesticus when presented with the choice of opposite sex stimuli of Domesticus and Musculus. However, at the border of the hybrid zone, we have associative mating both in Musculus and in Domesticus, and this is much more strong in one side of the hybrid zone. And the hybrids present a peculiar pattern that I'm not going to detail here, which is most of the hybrids present a preference for Domesticus, and only a small part of it present a preference for Musculus. So we were here in a condition where selection against hybridization occurred, and we find this pattern, geographical pattern, and we deduced that probably it is reinforced. However, competitive interference could occur in the hybrid zone in Denmark because our two subspecies are not very much different in terms of their ecology, and we know that domesticus male dominate domesticus musculus males. So there could be a matter of competitive interference influencing the evolution of signals in this system. So this is what we tested. We tested that in the lab. So we used aquaria in which we simulated territories. So each mouse was set in an aquarium that contained soil bedding that it has marked for several days. And this aquarium was connected to the cage of the male-female, and they could connect through small holes in the wall. So this simulated a territory for each of the mouse. We habituated to this apparatus for 30 minutes, and then we connected the territories, and we let them interact for a maximum of 20 minutes, and we stopped the interaction if aggressiveness was too high. So what we tested here was what was the outcome. Was it different between domesticus and musculus, which one dominated, and the intensity of the aggressive interaction. And we did 4 different experiments, 2 that involved inter-subspecies encounters, one between contact mice and another between allopatric mice, and 2 encounters that involved intra-species encounters between contact and allopatric mice of the 2 subspecies. So if agonistic character displacement occurred, we predicted that encounters between contact mice should give different results than encounters between allopatric mice. And what we found is that as far as the outcome in the interaction went, domesticus dominated significantly all the encounters, and the pattern wasn't different between allopatric trials and contact trials. So we concluded that there was no agonistic character displacement, at least on dominance. And we compared the intensity of the encounters, and an index of this intensity is the attack latency, and it is negatively correlated with the agonistic the intensity of aggressive behavior. And so what you find here is that that latency to attack during contact trial is much lower than during allopatric trial, much lower. So, it is significantly lower. So we could say that there is probably some character displacement, at least on intensity. So if the signals that we were studying during mate choice evolved in a competition context, we would expect that these signals, we would expect that these signals would allow subspecies discrimination. And the only way for us to disentangle between evolution in mating condition and competition condition was to compare whether the mice could discriminate each other, whether a mouse could adapt its behavior to the identity of the opponent. So we compared behavior of the mouse when he met another species versus when he met a mouse of the own species. And we expected that discrimination would occur if this happened, and that it would be stronger in the contact than in the allopatric paradigm. So what we found is that it didn't differ, and here I present a selection of the results. In yellow, you have the behavior during interspecies encounters, and in red, the behavior during intraspecies encounter. And what you can see is that the yellow and the red is not significantly different. There is something where the red, the intraspecies encounters are much more variable than the interspecies, but they are not different. And also, the pattern in the contact mice is not different at all from the pattern in the allopatric mice, which led to conclude that there is probably no evolution of a signal during a competitive context that could be involved in the pattern that we have been evidencing in the context of the camp. So this might be a bit complicated, but we have an example where this was shown, and this is very evident. And this is the example of the interaction between collared flycatcher and pied flycatcher. So this is one of the first examples of reinforcement in a vertebrate. And what this author found is that in sympatry, there are different forms of different morphs of male pied flycatcher, and in sympatry, this morph is much more frequent. And this morph resembles very much the one of the female collared flycatcher. And what they found is that this allows the flycatcher to avoid very costly competition by the collared flycatcher. So this signal evolved in a competition context, and is also used by sympatric female that prefer males that have this morph. So in the case of the house mouse, this doesn't seem to be the case. There is some character displacement, but it doesn't seem to be influencing species recognition. However, the reinforcement hypothesis seems to be still strong in that case so far. Thank you.